Good afternoon, everyone. I'm Chris Bray from the Clinical Trial Service Unit at the University of Oxford, and I'd like to welcome you all to this latest lunchtime webinar hosted by the MRC's HTMR Trial Conduct Working Group. The aim of these webinars is to help members of this group and other staff working the MRC network of hubs to share with a wider audience information about their projects related to trial conduct. This webinar has been promoted across the MRC network of hubs, the UK Trial Managers Network, and the UK CRC Network's Efficient Trial Conduct Working Group. And I believe we've had some attendees from outside the UK with us for the first time today too. We've got a good number of people online and we'll be getting started very soon. Please note that today's webinar will be recorded and should be available on the MRC HTMR Network website in about a week's time. So it's now my pleasure to introduce Jemma Hopewell, our speaker today. Jemma is an Associate Professor and Senior Scientist in Clinical Trials at CTSU here in Oxford. She has been heavily involved in several large-scale cardiovascular clinical trials over a number of years and is currently leading the statistical team on REVEAL, an international cardiovascular trial that has randomized over 30,000 participants. One of Jemma's research interests is in approaches to improving trial conduct, and today she'll be giving us a brief overview of some of the work she and her team have been doing on central statistical monitoring. Jemma's presentation will probably last around 25 minutes, following which there'll be an opportunity for you to ask questions or to make comments. If you're not familiar with how to do this in WebEx, we'll be showing you a slide to provide guidance at the time. So, over to you, Jemma.
Thanks, Chris, and good afternoon, everyone. So, as Chris has said, I've been asked to give you a 20 to 30 minute whistle-stop tour of some of the work that's being undertaken at CTSU in relation to improving trial efficiency, and particularly in the context of central statistical monitoring, and give you some of our experiences from our large-scale cardiovascular trials. In terms of an overview of what I'm planning to cover, firstly we'll have a look at the motivation and guidance for quality assurance in trials, look at the role that central statistical monitoring can take in this, and then I'll describe to you some of our hypothesis-driven and the more hypothesis-free approaches we've taken in one of our cardiovascular trials, and the results of some of the on-site validation. So firstly, what is our motivation? Why do we really need reliable evidence from clinical trials? Well, obviously, trials evidence is essential for appropriate decision-making in relation to the possible benefits and potential risks of clinical interventions. And a trial that's poorly conducted is really quite likely to generate spurious and unhelpful results, rather than any helpful information. Furthermore, clinical and regulatory decisions, in the absence of reliable evidence, for whatever reason, be that because they have never been performed or they've been performed badly, may actually harm patients and wider public health strategy. So it's really quite clear that there is strong motivation for the need to conduct trials well, and this is where monitoring may be really quite a useful tool for us. Monitoring is recommended by GCP and is widely used across a full variety of clinical trials in an attempt to identify potential problems. Monitoring can take multiple forms, can include oversight by trial committees, for example, on-site monitoring, data checks, and central statistical monitoring, or even a combination of all of these things. Historically, the main focus has been on on-site monitoring, and this has been the main strategy adopted. However, recent FDA draft guidance now encourages greater reliance on centralized monitoring practices than has historically been the case, and therefore less emphasis on the on-site monitoring. So we see this as a positive move forward. In terms of monitoring, firstly, obviously, we need to identify what the objective of an efficient and meaningful monitoring strategy is. Well, obviously, our major objectives are really to protect the participants, their rights, their safety and well-being, and ensure that the trial really generates meaningful results. And to do this, we need to make sure that there is good adherence to protocol. As well as these more obvious objectives, monitoring can also be very helpful in providing information that can help us focus further training and also help us to understand how to improve our study processes, not for just the current trial, but those in the future as well. The CTTI, the Clinical Trials Transformation Initiative, which strives to make clinical trials more efficient and quality-driven, recommends that we build this kind of quality into the design and conduct of trials by really focusing on what matters to us, by developing quality management plans and focusing performance assessments on important, meaningful parameters rather than data points and the like. And we hope to use this kind of information to improve training and other procedures. And they also encourage us to report the findings of our various approaches and how our quality management has gone during the course of a trial. So what is really important? Well, of course, we all are familiar with the concept of proper randomization. No foreknowledge of likely treatment allocation. We want to minimize post-randomization withdrawals and minimize potential loss to follow-up. In addition, obviously, we need sufficient relevant clinical outcomes for a trial to be useful. We want unbiased ascertainment of study outcomes. And these are generic factors that are really important to probably all trials and that will really impact on the quality of our trial. But there may be others in your specific study that may also be important. If we want to focus your monitoring strategy on examining data that describe the factors that are most important to your trial, including measures of patient safety, study reliability, and those that may identify important protocol violations. Remember, monitoring should avoid undue emphasis on data points, and a reliable result does not necessarily equate to accurate data, and vice versa. Furthermore, given these multiple objectives and possibilities, it's difficult really for a single monitoring strategy to be a good fit for all trials. So we really need to focus on tailoring our approach to what's important in the specific context. So why do we need to improve if historically on-site monitoring has been used? What are the problems with this? Well, this is a largely retrospective process, making it more difficult to find and implement solutions to any problems that are being identified. It's also commonly poorly focused, with the emphasis on document verification rather than important aspects such as compliance. And given that many trials have a large number of sites, it carries a really quite large financial burden and simply isn't practical in most instances. Given this, new guidance really recognises these problems and has now come to the opinion of moving towards more risk-based monitoring, in which high-risk sites are identified in order to guide on-site monitoring activities. And this is where essential statistical monitoring can become important and a really useful tool to us. This change in guidance from the regulatory authorities has really created a bit of a boom in the last few years in terms of new literature looking at various approaches, spanning a whole plethora of potential approaches to undertaking central statistical monitoring. So, how are these different types of central statistical monitoring useful, especially in multi-centre trials, which I'll concentrate on? Well, there are a variety of data issues that could be present in any study and that we'd really like to know about. So, particularly we'd like to know about fraud, whether data have been intentionally falsified, so potentially modification of eligibility criteria or something similar. We might also be able to find evidence of tampering, where data have been knowingly fabricated, such as propagation of results from previous visits. For example, the blood pressure measurement from a year ago may be carried forward to the current visit. Sloppiness, which can commonly mean that the protocol is not being followed accurately and may lead to problems such as underreporting of events, which of course is extremely important in terms of making sure that our trial is useful. And finally, errors, such as equipment problems or failures, where there's no intention to mislead the trial investigators, but that all the same are important in our measurements. So given all these various data issues, we can use central statistical monitoring to identify unusual sites and thereby help to prioritise on-site visits and additional training, generate actionable objectives throughout the trial, and ensure overall data quality. The main approaches that we've used, especially in CTSU, are a hypothesis-driven approach focusing on key risk indicators, which are basically a small number of variables that are important to the study and its ability to produce reliable results, but also looking at hypothesis-free, more database-wide approaches, such as the SMART software developed by Mark Boyes and team, that really look at all the data that's being collected. So let's look at an example. HPS2 Thrive is a large cardiovascular, double-blind randomized trial involving patients with prior vascular disease, and this trial randomized over 25,000 patients to extended-release niacin lorograpant or placebo, with all patients being on a background of standard statin therapy. So patients were on the randomized therapy for around 4 years, with follow-ups conducted at least every 6 months. And this trial examined the effects of niacin on major vascular events, and the results of this study were first reported last year and more recently in the New England Journal of Medicine. When collecting the Thrive data, visits were mostly conducted in clinics with either a nurse or a doctor, and these took place in 3 regions, UK, Scandinavia and China, and combined we had a number, 244 sites, and roughly a third of the sites were in each region. The average number of participants varied considerably per site, but the average was around 100, albeit slightly higher in China. The data collection procedure was via electronic case report forms, which were then promptly synchronized with a central database held in Oxford. These electronic data capture methods enabled us to record patient blood results that were undertaken at each study visit, general visit information, such as anything the patient was telling us during the visit, as well as adverse event data that was required. However, obviously this trial is based in 3 different regions, and there were some differences between how the protocol implementation was adhered to. Particularly, for example, Chinese patients were seen by doctors and UK patients were seen by clinic nurses. And so what we needed to do was kind of account for these differences that were perfectly reasonable by stratifying by region in our central statistical monitoring analyses. So as I indicated, our main approach in Thrive was to use KRIs, and this focused on a relatively small number of key variables. In Thrive, we formed a monitoring team based on statisticians, principal investigators, and the International Monitoring Coordinator, and the KRIs were selected by this committee by considering the variables that were available to us and their generalisability, so variables that were of interest to all trials, such as the rate of adverse events reporting, things that were important to us as a specific trial, such as essentially muscle symptoms in statin trials. We considered the availability of data and also our experience from previous studies and our on-site visits over the years. So we could incorporate all this information in generating what we believed would be a list of meaningful key risk indicators. The basic strategy with KRIs that we developed was firstly for each KRI to create a summary measure of interest. So for each site, a summary measure such as a mean, variance, a proportion, or something like that was calculated. So a good example of this might be the serious adverse event reporting rate. Secondly, we wanted to identify extreme sites, and to do this, we defined a KRI-specific critical threshold, and sites that exceeded this threshold were given a score of 1, and those that did not were given a score of 0. So, for example, this gives sites with a low SAE reporting rate, which was considered to be problematic to us, a score of 1 relative to comparable sites. And again, emphasizing that these needed to be comparable sites, so they were in the same region. In the example of Thrive. These first 2 steps were then repeated for other KRIs of interest, and then finally we produced a summary score based on all the KRIs examined. And this is based on the sum of the individual KRI scores, weighting each KRI as considered appropriate, giving higher weight to the more relevant KRIs or more important things as we saw fit. So how does this work in practice? Here's an example in HPSC Thrive based on the reporting of serious adverse events. This is considered a particularly relevant key risk indicator, as underreporting of serious adverse events may indicate poor adherence to protocol and also can impact on the study power. So we defined a critical threshold, but particularly wanted to identify sites that were underreporting SAEs. And a centre was considered extreme if the SAE rate in a particular centre was less than half the median of the overall SAE reporting rate in comparable sites. In the table on this slide, you can see the related characteristics for 4 of our sites, and particularly those reaching this threshold. The median serious adverse event rate in the UK, for which these centres are relative, was 0.3. And as you can see, our 4 sites, the annual rate of reporting was all less than 0.15, which is half that overall median. It also helps to be able to present this kind of data graphically. And here we see the results for all 3 regions, with each point representing a site, and larger values on the y-axis indicating more extreme results from the site. The coloured lines indicate strata, so in this case this is region specific, and you can see quite clearly a small number of extreme sites, and that the threshold differed between these different regions. Combining the work that we've done on serious adverse event rates with other KRIs of interest, such as short visit duration, which we considered to be particularly worrying, given that the required forms couldn't be filled out in a very short period of time, and therefore was indicative of the protocol not being adhered to. We also looked at extreme distributional properties of safety biomarkers, so mean, standard deviation, kurtosis, and skewness. We looked at non-compliance, obviously very important to lots of trials. And we weighted these as considered appropriate, and that's why you see values in the table that are less than 1 for the individual KRI scores. Given these various KRI results, we then summed up the individual KRI scores to create the final combined scores on which to rank the site, as this is then used to prioritise our on-site visits. When we attempted to validate the results of our KRI analyses using on-site visits, we found that all the sites with a high KRI score seemed to have some underlying problems, mostly just poor performance, but things that we would like to action upon, for example, provide additional training. On the reverse side of that, we saw that sites that had low KRI scores rarely had problems, suggesting that our KRI approach was really picking up what we wanted it to, and identifying the high-risk sites. Doing this enabled us to detect issues, not just those from the KRIs, but also to visit those sites which commonly had small additional problems, and allow us to put remedial actions in place. And overall, this validation suggested that on-site and central statistical monitoring are really quite complementary approaches, with both required to produce an efficient and effective monitoring schema. However, our approach was very specific to our trial, and there are some limitations. The method that we developed for use in Thrive is generally not suitable for longitudinal key risk indicators and looking at changes over time. The critical thresholds that we defined were somewhat subjective and really based on expert knowledge rather than a predefined rule, and therefore not easily generalisable to the wide variety of trials. The binary score that we give each KRI to identify an extreme site really ignores the extent of deviation from the other comparable sites. So we can't identify which site was just slightly more extreme than the rest versus a site that was very much more extreme than the rest. And finally, it really relies on site size being of a reasonable, maybe at 20 patients or so. And commonly, especially in smaller trials, we may find that sites are much smaller than 20 patients. But these kind of sites still require monitoring. Given this, we're now moving on and we're working on generating a new, more generalisable approach. And our preliminary findings suggest that the new improved method that we're working upon will be particularly more useful for the wide variety of trials out there. In particular, it should be more generalisable, also suitable for multivariate correlated key risk indicators as well as normal univariate KRIs. It will allow for smaller sites to be included in the analyses, and also for this site size to be accounted for in some way. It also will provide a p-value threshold rather than a subjective threshold defined by the monitoring committee. And also the final summary score is p-value based, making it easy to rank sites, but also being able to assess to some degree the magnitude of how extreme a site is. However, the warning sign for this really is that it is much more statistically complex, and there are some advantages and disadvantages, and we need to balance these up between more simple and more complex approaches in terms of usability, because a monitoring approach needs to be easy to use and not to place too much requirement on the central coordinating centre. Finally, I wanted to mention another method that we've also applied in HPS 225, which has been done in collaboration with Mark Boyd and his team. And this involves something called the SMART approach, which, in contrast to the more hypothesis-driven KRIs, is a hypothesis-free approach, analysing all available variables and really considering them all to be of equal importance. The advantage is that we can check for aspects that are not captured by the KRIs, and this can enable us to look at overall study reliability and not just those in a specific context. So, for example, SMART would look at variables that we may not think relevant, but actually may be proxies to things that are relevant, and thereby find hidden data problems that wouldn't be found by a KRI approach. The SMART software compares each site to all other sites using a variety of statistical tests based on means, measures of global variability, looking at proportion of outliers, also looking at dates, so for example the proportion of visits that took place on a weekend, which obviously isn't conventional in a 5-day working week, and looking across the whole of a trial database runs multiple tests per variable. And therefore, the statistical test that it generates creates a very large number of associated p-values. So, for example, you may generate more than 100,000 p-values for a trial with 10,000 participants and 100 variables. So this very quickly becomes a large data problem. Given all these different p-values, SMART then enables you to generate a summary score and providing an overall ranking and an ability to identify the most extreme outlying sites. Here's an example of the distribution of the summary score produced by SMART during the course of HPS 225. And as you can see, the most extreme sites are highlighted, with the most extreme site being UK 75 at the far right hand of the plot shown. The SMART version used in HPS 225 is now, there are now newer versions available that have additional features, and those are available by speaking to Mark Boyd directly. In addition, this is an example of the output from SMART, and what is shown here is the output for a particular site, Centre UK 75. It shows the overall score provided and the rank compared to all other sites, and of course the number of patients that are in that centre. As well as the summary score, it shows the individual tests that have been conducted and the individual p-values, also giving the individual ranks per variable and test. Negative p-values here represent when an under-reporting or a lower value than you would expect in the remaining centres. One contrast to this, to SMART versus the KRIs, is that SMART doesn't worry about direction. So in the KRI approach, we look at whether SAE reporting rate is lower than normal, because this is particularly important to us. But SMART would consider a low or a higher rate of SAE reporting equally important. In terms of comparing the kind of more hypothesis-driven versus the more hypothesis-free approach, here you see a table of basically the key advantages and disadvantages. KRIs are tailored to each individual trial and therefore offer a relatively easy to interpret result that's really useful for your specific trial and enables you to identify variables to which you can attribute actions within the on-site visit. However, the current approach that we've used, the scoring and criteria are somewhat subjective, but hopefully in our newer method that we hope to describe to you in the next few months, some of the subjectivity would be eliminated. And finally, the KRIs allow for variable weighting that's quite flexible, allowing you to really use your prior knowledge and expertise to appropriately attribute what is a high-risk centre. In contrast, a hypothesis-free, SMART, or there are other similar approaches, provides an unsupervised method requiring very little expert or background knowledge of the study. However, stratified analyses are not generally implemented, albeit they can be done manually. They make specific database structure assumptions, which might not be the case for your specific trial, although with a new CDISC standards, this will hopefully become increasingly easy. And also, it weights all variables, and particularly all statistical tests, equally, which may not be what would be most advantageous. I think our general conclusion from having looked at two contrasting approaches is that actually they both generate some complementary and useful results. So similarly to the on-site monitoring and KRIs and SMART are considering all this information together is actually probably the most powerful tool. So at the end of the day, what are our conclusions? Well, given our overall objective, which is to improve the availability of reliable information from clinical trials on which we base our healthcare decisions, it seems that we can undertake monitoring that can enhance the quality by identifying threats to design, performance, and analysis. But our approaches to monitoring should be appropriate to the setting, proportionate to the risks, stimulate improvement and further research, and this will then enable us to mitigate the risks to participants and to the reliability of the results throughout the trial. And I believe that our various approaches to statistical monitoring can really facilitate this. Finally, I'd like to acknowledge the team working on this, particularly Dr. Elsa Valdez-Marquez, who's doing much of the day-to-day work on this, and our other colleagues such as Jane Armitage, Martin Landry, and Carol Knott. Our colleagues and collaborators at SMART IDDI, David Vanette, Eric Bafana, and Mark Boyd, and obviously our funding sources. And if anyone wishes to contact me by email, then my email is provided at the bottom of the page. We're now going to start our question and answer session, and here's a slide to tell you how to do that. If I hand back to Chris.
Okay, yeah, so if you could keep that slide up, please, Jemma, so that people who aren't familiar with ways in which you can ask questions in WebEx webinars have some guidance. Essentially, you've got two methods there. If you are connected via audio, you have speakers and microphone either through your computer or your phone. then if you raise a hand, we can invite you to put a question or comment to Jemma. Alternatively, you could type in a question to us in the Q&A panel, and we'll read that out on your behalf. So just while you're getting your heads around that and maybe thinking about questions to put to Jemma, I'll just get things started. So Jemma, you talked about the KRI approach and the fact it was developed in the context of HPS2 Thrive specifically. Have you been successful in applying that approach to any other trials yet?
Well, we're using the same KRI approach in the HPS3 REVEAL study, which again is a large-scale cardiovascular study, 30,000 patients randomized, and sites internationally, China, the US, and various sites across Europe. And actually, we've been quite successful again in using the same approach to identify sites that are troublesome. And we have particularly identified sites where further action was needed. And I think the advantage of being able to do this really is that we're running these KRI analyses on a 6-monthly basis as new follow-up data come in, and therefore we're able to subsequently follow up with a centre within quite a short period of time and put fixes into place, rather than, in contrast, maybe the old style approach, which is more waiting to the end and then hoping nothing went wrong during the course of the study. So yes, I think it is generalisable to large-scale cardiovascular trials, but I think the newer approaches and hopefully more generalisable ones that we're working on will maybe be more useful for the general trials community.
Okay, thanks, Jemma. We've had a few questions typed in, but we think these are by people who might also have audio. So I'm just going to see if Marianne Thompson has a question that she'd like to put to you. So if we can find her and unmute her microphone, hopefully she can ask you directly. So Marianne, hopefully you're now in a position to be able to speak. I'll put your question directly to Jemma.
My question is how to move this method for smaller trials with field size.
Did you catch that, Jemma?
No, I'm sorry, the sound quality is quite poor.
Sorry, could you repeat the question slowly? We had a bit of a problem with the sound quality then.
Jemma, how to move this method for smaller trials with field size?
Okay. So the question is relating to how useful these methods are for potentially smaller studies, but also for those with fewer sites. So these methods have been developed in the context of having, first of all, a relatively large number of sites with which to compare. And obviously, when you're looking at multi-centre trials, you want to compare with other information. The more information you have, obviously, the better. But I think these are perfectly applicable to studies where there are maybe many fewer sites than we have in HPS2 Thrive, for example. So maybe you only have 50 sites. And the reason that we've actually looked at this a little bit, because obviously we've developed stratified analyses, and I think that in that context this is fine. In terms of the second part of your question, which relates to how small the actual centres are themselves, as I said, the approach that we've adopted within Thrive really kind of relies on about 20 patients within each site. But the newer methods that we're trying to develop because we do realise that many trials may have smaller sites, we'll be able to take this into account. I think as long as you've got about 20, then that's kind of a general rule of thumb.
Okay, thanks, Jemma. I think we're now going to ask Norman Williams from UCL if he would like to put a question directly to you. Norman, we just unmuted you. Hello, it's Norman Williams there. We think you have a question for Jemma. Please go ahead. Maybe not. I can read out the question, Jemma, if that helps. So Norman says, can you use KRI scores and cut-offs from a previous similar study? This would enable the method to be used from day one so that sites requiring training can be identified rapidly.
I think the answer to that is dependent on your context, of course, how similar the trials are, but indeed in implementing some of the strategies within the eCRF3 reveal trial, we did use information that we generated from eCRF2's trial. And I think that's really quite sensible, especially if you're in very similar populations, and you really believe that they are comparable in the context that you're describing. And as you say, and as Norman says, it's particularly helpful in the early stages of a trial when maybe there haven't been enough patients generated per site, or enough sites to compare more reasonably. So I think general answer to that is yes, with a few caveats.
Okay, we've still got quite a few questions to choose from. Matt Sides has typed a question in, but I'll just see if he's available to put that to you directly to make things a bit more interactive, possibly. Matt, we're going to unmute you. If you're able to put your question to Jemma, please go ahead. Right, that tactic isn't working. So, Matt says, lovely presentation, thank you. How long into a trial must you wait before you can first start to apply these methods?
It's a good question, and it will always depend on your rate of recruitment and on the particular trial that you're looking at. Obviously, you need sufficient data to be able to make the comparisons, and that's probably where Norman's strategy probably works quite well in terms of at least defining critical thresholds. It also comes about when you're thinking about how often you want to repeat your central statistical monitoring analyses. And really you want to make sure that you're generating enough sufficient new data such that it isn't totally outweighed by the historical data. And some of the ways we actually do that is by looking at kind of most recent visits, maybe within the last 6 months, to make sure that we're picking up new signals rather than something that's happened historically and actually may have been fixed in the interim. So I don't think you can actually put a definite number on it, but you can pretty much start as soon as you've got enough patients into your study where you consider it statistically sensible. You can compare that to any other statistical concept where, too few observations equals an unreliable result.
Thanks, Jemma. We're going to have to leave it there, I'm afraid, but thanks also for offering to answer any additional questions by email. So thanks again to everyone for joining this webinar, and especially to Jemma Hopewell, our speaker today, for a great presentation and for fielding your questions. There are just 2 points I'd like to make before we wrap up. Firstly, as I mentioned earlier, a recording of today's webinar should be available on the MRC HTMR network website in about a week's time. Just go to methodologyhubs.mrc.ac.uk and follow the link to webinars. You'll also find here recordings of the previous Trial Conduct Working Group webinars, including Sally Stenning's webinar from October, which also related to statistical monitoring approaches. Secondly, my final point is we will be arranging further webinars relating to trial conduct research during 2015. Most likely these will be on the first Tuesday of each month, and they will be promoted through the usual channels, so please look out for further information early in the new year. Thank you and goodbye.